Estimation of total serum protein by Biuret method. The method that we will be doing in the lab is known as Biuret method. The reagent we will use is the Biuret reagent. Okay. Before that you did estimation of glucose by what method? Godpot method. Okay. So what is plasma protein? Blood contains large varieties of proteins. The major ones are albumin, globulin, fibrinogen. Other examples are proteoglycans, metalloproteins, lipoproteins, and etc. So the major ones are what? Albumin, globulin, and fibrinogen. Then proteins are synthesized mostly in the liver except immunoglobulins and hormones. In what type of glands are hormones synthesized? Endocrine gland. And this immunoglobulins? White blood cells. WBCs. Okay. So functions of these proteins are they act as enzymes, hormones, clotting factor, transport agent, protective agent, nutrient agents, immunity. So this is what all the things the protein does. Various functions. Okay. So maintenance of the colloidal osmotic pressure is done by albumin. Do you know what is colloidal osmotic pressure? So in our blood vessel there is a pressure known as the colloidal osmotic pressure. If the pressure is absent, then our blood vessel will collapse like this. Normally our blood vessels have this much gap, right? So that the blood cells, fluid or various substances can free flowly travel. So to keep it from collapsing, albumin plays the part. Okay, albumin plays the part. It helps in resisting the blood vessels from collapsing so that the substances inside the blood vessels can have a free flow movement. Okay, that pressure is known as colloidal osmotic pressure. Then maintenance of acid-base balance, that is the buffering action of protein. So if your body has more H plus ions, what is the state known as? Acidic state. So what the protein will do, it will go and bind with the H plus ions. Okay, then the concentration of H plus ions will decrease, right? So in that way the body will come to a normal state. Similarwise, if it has more OH minus ions, then it will be alkaline, basic alkaline, same thing. So again the protein will go and bind with the OH minus ions, and in that process the concentration will again decrease and the body will come to the normal state. So this is what we say that our body copes up. This is the coping mechanism. This is how by the buffering action of protein the pH of the body is maintained. Okay, by the buffering action of protein. Then transport mechanism, that is albumin, alpha globulin and beta globulin. So can you tell me how does the bilirubin travel from the blood to the liver? By binding with albumin, right? So now you know how they help in transport. Okay, then defense mechanism, that is gamma globulin. Now defense mechanism means what? It helps your body fighting against infections, cuts. When there is cut, it goes and helps in fighting with all that. So this globulin is one of the major constituent of the protein, and again globulin has subtypes, that is alpha globulin, beta globulin and gamma globulin. So gamma globulins acts as defense mechanism, helping your body fight against infection. Then blood coagulation, that is fibrinogen. So fibrinogen is one of the 12 clotting factors. Okay, we have 12 clotting factors and fibrinogen is one of them. So when you get a cut, it bleeds. But after some time the bleeding stops and there is some dried clotted blood on top of the bleeding, right? So what happens is fibrinogen goes and gets deposited over the cut, and along with other blood cells like RBC, WBC goes and gets clotted over the cut area, and then it stops the bleeding. Okay, and that's what you see, those dried blood you see. After some time, those are the fibrinogen along with other blood cells. Now, what is plasma protein? Plasma protein is albumin, globulin and fibrinogen combined. All three combined is known as plasma protein. What is serum protein? What is serum? Plasma protein without the clotting factor. So, if you take a sample of blood and you put it into, if you centrifuge it, what will you get? I'll get the serum. The supernatant portion, the supernatant liquid is the serum, and all the components and this fibrinogen and the clotted things will be sedimented down below, right? So why fibrinogen is not there in serum? Why this serum protein is plasma protein minus fibrinogen? Yes, because it gets clotted. It itself is a clotting factor, right? One of the 12 clotting factor is the fibrinogen. So it settles down, gets clotted and gets settled down when you centrifuge the blood. Okay. Then the normal range. Total protein ranges between 6 to 5 to 8.5. Now in some places you can get 6 to 8 as well. Both are correct. 0.5 difference, doesn't matter. Then serum albumin is 3.5 to 5 gram per dl. Serum globulin is 2.5 to 3.5 gram per dl. Fibrinogen is 200 to 400 milligram. Don't get confused. These are in gram and this is in milligram. Fibrinogen is 200 to 400 milligram per dl. Now albumin globulin ratio is ranging between 1.2 to 1.2 is to 1 to 2.5 is to 1. These are the normal values. If the albumin is less, if serum albumin is less, will the Ag ratio be more or less? Increased or decreased? It will be decreased. And if the globulin is less, then Ag ratio will be then increase. Did you understand? If albumin is low, then the Ag ratio will be low. And if globulin is low, then Ag ratio will be raised. Albumin globulin ratio means what? Albumin divided by globulin. Okay, albumin divided by globulin. Now if you take any ratio where the serum albumin is suppose 3.5 to 5, suppose you take it 2. Now 2 divided by normal globulin may be 3. 2 divided by 3 is how much? 0.6. So is it normal? Is it normal? 0.6 is not normal. It is decreased, right? So the Ag ratio is decreased. Did you understand the maths what I did? Now suppose you take albumin 4 and globulin you take 1, 2.5 to 3.5. So you take 1. Then what will be the this one result? 4. So is it normal? No. So now you understood. If albumin result is low, if albumin is low, Ag ratio will be low. If globulin is low, Ag ratio will be increased, high. Both are abnormal only. Okay. Now what is the principle of Biuret method? All compounds containing 2 or more peptide bonds react with cupric ions in alkaline medium to form a purple color complex. What was the color in your glucose Godpot method? Pink. Over here you will get purple color. So protein is not a single molecule. It has multiple amino acids. Now all these amino acids bind with each other with peptide bonds. Now for your test to come, you need more than 2 or more than 2 peptide bonds. So if you have only 2 amino acids, you will have only 1 bond, right? Only 1 peptide bond. This you will not get the result. So you need to have more than 2 peptide bonds. All compounds containing 2 or more peptide bonds react with cupric ions in alkaline medium to form a purple color complex. These cupric ions and alkaline medium is provided by the Biuret reagent. Okay. So the absorbance of the complex is measured colorimetrically at 540 nanometer. How much at what did you measure the this glucose? 520. In the calorimeter you measured it at 520. In this protein estimation by Biuret method you will measure at 540. Now the kit that you are using is the Corel kit. So according to the Corel kit, it says you have to measure it at 550. 550. Can you see 550 over here? Yes. 550. It says 550. Now we don't have 550. In the colorimeter that we are using, we don't have 550. So we'll measure it at 540. So the absorbance of the complex is measured calorimetrically at 540. Clear with the principle? Now what are the reagents that are required? In the Biuret reagent that we are using, we have there copper sulfate. Copper sulfate, that is the source of copper ions. Then we have sodium potassium tartarate that stabilizes the copper ions. Now, so that to avoid hyperreaction, this sodium potassium tartarate stabilizes the copper ions. Copper ions was sourced by copper sulfate. Okay, copper sulfate is the source of copper ions, but to avoid hyperreaction, sodium potassium tartrate stabilizes the copper ions to avoid the hyperreaction. Then we have the potassium iodide that acts as antioxidant. What is antioxidant? What is the meaning of anti? So what is antioxidant? Yes. So potassium iodide acts as antioxidant and sodium hydroxide provides alkaline medium. Don't get confused during Viva. In Rotherals, what provided the alkaline medium? Liquor ammonia. In Biuret, sodium hydroxide will provide the alkaline medium. Then we need standard protein solution. The kit that we are using, the Coral kit, and as well as during your exam you will get the same kit. The standard protein solution is 8 gram per dl. But in other kits, there are many kits. So in certain other kits you can get 6 gram per dl as well. So it depends on the kit. Whatever you are using, whatever the standard is mentioned on the kit, you have to multiply with that. In glucose, how much did you multiply with? 100. Over here you will multiply with 8. Then you need test serum and distilled water. So the procedure is same. You will take 1 ml biuret reagent and in the blank you will add 20 microliter of distilled water, right? In your Godpot method, how much distilled water did you take? 10 microliter. Over here you will take 20. Okay, and 1 ml or 1000 microliter biuret reagent. Okay, then next you will take 1000 microliter biuret reagent and 20 ml of standard. Okay, then in the third one you will take 1000 microliter of biuret reagent and your test, whatever sample you will be provided, you will take 20 microliter of the test. Okay, it's the same thing, only the ratio has increased. Okay, so the observation, after you take your reagent and your after you prepare the blank, standard and test, you have 3 test tubes. You will mix them and then you will put it into what? Incubator for how many minutes? 10 minutes. At what temperature? 37 or room temperature. So whatever reading you will get, you will put it here, blank, standard and test. You will get 3 readings, right, after 10 minutes. So you will note it down like this. So this is the calculation, this is the formula. Total serum protein concentration in gram per dl equal to ODT, that is your OD test, minus ODB, that is your OD blank, by ODS, that is your OD standard, minus ODB, that is OD blank, into concentration of standard. Now the concentration of standard was 8. The formula is similar to your Godpot one, right? ODT minus ODB by ODS minus ODB into concentration. So over there you multiplied with 100, over here you will multiply with 8. Now according to the kit, there is another formula to do it. In your last class also, for glucose estimation also you did, right? ODT by ODS into 100. Did you do it? Yeah. So over here also you can do it, because see, according to the formula given in the kit, over here, can you see absorption T by absorption S into 8. Okay. According to this formula also you can do, but did the result come similar or less than 10% difference was there in your Godpot method? Did it come, or did it come, there was a vast difference? Do you know how to do the procedure? Do you know the procedure, what you have to do? Yes. So you have to take 1000 microliter and then in blank you will take 20 microliter of distilled water, and in test you will take 20 microliter of the standard solution, and in the test you will take 20 microliter of the whatever serum or whatever is provided over there. So after that you put it for incubation for 10 minutes, you bought it. Now what will you do? How will you check the absorption in the colorimeter? What do you have to do first? How will you bring to the optimum this one percentage to, how will you do it? Set the colorimeter in 0 using distilled water. More than 3 ml of distilled water you will put in the cuvette and you will set the machine into 0. Okay, for calibration, for optimum calibration. Okay, after that, if you are using this formula, after that you will take your blank, you will pour it in the cuvette, you will take the absorption value, you will note it. Again you will take the test, you will pour it in the cuvette, you will take the absorption value from the colorimeter, you will note it, and again you will take the blank, then standard you will take the absorption value and you will do it, and then you will put the all the ratings, whatever result you got, you will put in the formula, multiply with 8 and you will get the result. But do you know how to do the other one, ODT by ODS into 8? Do you know how to do it? Didn't you do it? Yes. In this procedure, when you are manually subtracting ODB, you are doing it with your manual calculation. But when you are using the other formula, that is ODT by ODS into 8, over there you will subtract the blank with the help of the machine. The colorimeter will do it for you. You will take the standard, you will take the distilled water, that is more than 3 ml distilled water, like you took in this formula, you will put it in the colorimeter, you will bring it into 0 for optimum calibration. You did it. After that, next you have to take the blank, right? Now when you will put the blank in the colorimeter, you will get certain finding. What is that absorption finding that you are getting? Is it of the Biuret reagent or the distilled water? Because blank has what? 1000 microliter of reagent, Biuret reagent, and 20 microliter of distilled water, right? So distilled water has already been set to 0. So whatever absorption value you will get after you put blank, that is of the reagent. That is only of the reagent. So you have to bring blank to 0 so that you can get the correct correction of the test and the standard in the division method. In this division method that I have showed you here, that is ODT. You can note it down. It is the second formula, ODT by ODS into concentration. This is the second formula. If you are doing it according to this kit manual, concentration of standard, that is 8. And when you do both the methods, when you will go there in the lab, some will do this one, this formula by subtracting manually, that is with your hand you will do the calculation and subtract. But if you are doing the other one, that is the division one, when the machine will subtract it for you and you will only do the division, in that also you will get the same value only. Now if it is, suppose somebody got this one 10 and somebody got 9.8, is it same or different? It is same. The difference is of what? 0.2. So it's same. I was saying that when you give your blood test in 2 different labs, do you get the same result? You won't. You will get 0.5, 0.2 difference will be there. And that varies because of the kit. Kit to kit gives you different result. But the difference is of not much. Not more than 10%. If it is more than 10%, then what is the mistake you did? The mistake was in pipetting, because in this particular test you will do, your pipetting matters, because if you are not doing the pipetting properly, that means that you did not take proper amount, because the amount itself is very less. 20 microliter is less than 1 drop. So if you don't do the pipetting properly, you will get wrong absorption values from the calorimeter, whatever you say. So if you get wrong values, ultimately when you will do the maths, doesn't matter with this formula or the second formula that I said according to the manual, the result will come more. If it is supposed to be hypoproteinemia, if it comes if it comes less than normal, or if it comes hyper, more than 10, is it normal? It's not normal. So it will give hyper, but abnormal value of hyper. That means it was wrong in your pipetting. So your viva will also go bad like that. That means you don't know pipetting. And in this particular test, pipetting should be very good, because calculation you can do. Calculation is very easy to do, and the main part is the pipetting. In this test, main part is the calculation is easy, but if the pipetting goes wrong, means the whole test is wrong. Then again you have to do the pipetting, wait for another 10 minutes and then do the calculation. Do you understand? So these are some important points to note. Other methods to estimate serum protein. There are other 2 methods. We are using the Biuret method. There are other 2, that is Geldal method. Okay, K is silent here. K is silent. It's pronounced as Geldal method. And Lowry method. The second one is Lowry method. Then causes of hyperproteinemia: dehydration or hypovolemia, chronic infection, collagen disease, multiple myeloma, macroglobulinemia. Why in dehydration do we get hyperproteinemia? What happens in dehydration? So you know the answer. So dehydration, the water concentration, the fluid in body is decreased. So why we will have hyperproteinemia at that time? What will happen if the fluid is decreased? Means concentration of the protein will increase. That's why in dehydration we have hyperproteinemia. Chronic infection. Then what is collagen disease? What is collagen? Skin. After you get a cut, then there is some thick what? Collagen fibers. When it gets healed, now someone will have a thin scar, some have very fat, very thick scars. Why is that? Because of this collagen disease. They have collagen disease because of which the scar is more than the normal size. It was not supposed to be that big, but it is more than the normal size. So people who have this collagen disease, they can have this hyperproteinemia. Then multiple myeloma is a disease of the brain. No need to know in details. In final year medicine you can learn multiple myeloma. It's an important topic, but not required now. Then we have macroglobulinemia. What is macroglobulinemia? Emia means in the blood. Macro means big. Globulin is a protein. So big protein in the blood. Then causes of hypoproteinemia. Liver disease. Why do we get hypoproteinemia in nephrotic syndrome? What happens to the liver in nephrotic syndrome? What happens to the liver? Is it functioning properly? Is it functioning? It's not. So what happens to the protein? Yes, the nephrons cannot reabsorb. Nephrons cannot reabsorb the protein back to your body, because of which you are losing the proteins through the urine. That's why in nephrotic syndrome you will have hypoproteinemia. What is hemodilutation or overhydration? What is hemodilutation? What is dilutation? Yes, more fluid. Or overhydration means more fluid in the body. So what will happen in that case? The proteins will get diluted. That will give us hypoproteinemia. Then malabsorption because of intestinal cause. If your intestine cannot reabsorb the proteins properly, that will ultimately result of less protein in the body. That is hypoproteinemia. Then extensive burns, that is lost through the skin. Because of burn, your capillaries can also get burned, and through that the proteins can ooze out. Then A-gamma globulinemia. A-gamma globulinemia is a congenital disease. What is a congenital disease? By birth. Disease that is present by birth. So this is absent gamma globulin. Absent gamma globulin in the blood is known as A-gamma globulinemia. Okay. Then we have malnutrition, that is kwashiorkor. Do you know what is kwashiorkor and what age group has it? Malnutrition? Childhood. Yes, children have it. Kwashiorkor disease. Then causes of increased AG ratio, hypogamma globulinemia. That causes increased AG ratio. I told you, right? If this globulin is less, then AG ratio will be increased, and if the albumin is less, then AG ratio will decrease. So causes of decreased AG ratio is nephrotic syndrome, because body cannot keep the protein inside, so it's losing. That is decreased albumin, ultimately leading to decreased AG ratio. Then we have chronic infection, multiple myeloma, which are the causes of decreased AG ratio. Okay. Bus. Okay. Slowly pour it. Okay. Then next who will take? You take. Not with this. And next you will take.
Whatever value you will show, always that after minusing the protein. For an example, let's say your body weight.
No, there's air. Wait. Yeah, okay, but don't release the pressure so fast.
Okay. OD is 0.30. Okay.
Next tube.
0.36.
Standard.
Now,
If you're doing standard, keep it in the standard. Yeah. Then next T.
0.36 minus 0.06. 0.30 minus 0.36. 0.024, right? But if you make this proportion as 0, whenever you are measuring the
Keep it. So BST done. Then you will take the blend. Same. Before doing it, you will check whether it is 20 microliter or not. Put the needle, small needle.
ODT will directly come as 0.36. Over here you
Take,
calculate.
oh, sorry, you will take this one. Standard distilled water.
Over here it's 0.34. So however you want
Okay.
to do it, the value is
So, yeah.
one thing.
Where is the test tube? Here. How will you put it? Very minutely. Put this one down very minutely. Go down. Yeah, release it now. Release it. Did you see the amount?
After your test is done,
Okay, done. Then we want to take this one or this one?
This one.
This one, okay. Take.
what is done, keep the attendance and everything. Okay? Tomorrow you have exam, I guess.
Okay, good.
Otherwise, if there is some other reason,
Maybe there was some amount of water there, that's why you did. No problem. We'll
I will just observe the cases. You all know how to do the purity, I will not be telling you. And I hope nobody will mistake it.
नीचे हाँ put it. Discard it hard. It will not come because it is made of egg yolk. That's why it's little thick. It's made of not egg yolk actually, egg white. The white portion, egg white. Okay.